Mycoplasma contamination can compromise cell culture quality and experimental results. This video shows how to use the Myco-Visible Mycoplasma qPCR detection kit from MP Biomedicals for routine screening and for various pharmacopoeia guidance. Results are available in about 2 hours. The kit detects Mycoplasma, Acoloplasma, and Spiroplasma species, including common tissue culture contaminants. It supports routine screening and testing workflows aligned with European Pharmacopoeia 2.6.7, Japanese Pharmacopoeia 18G3, and US P63. Before starting, prepare a clean workspace. Wipe surfaces with Myco-Out Mycoplasma removal spray, 70% alcohol or 10% bleach. Wear gloves, a lab coat, and eye protection throughout the procedure. The kit includes sample buffer, nuclease-free water, 2 times qPCR master mix, primer mix, internal control, and positive control. Store all components at -20 degrees Celsius. After first use, aliquot the master mix, primer mix, internal control, and positive control to reduce repeated freeze-thaw cycles. There are 2 sample preparation options. Use the routine screening workflow for standard cell culture checks. Use the extraction-based workflow for EP/JP or USP-aligned testing. For routine screening, transfer 1 milliliter of your cell culture sample into a 1.5 milliliter tube. Centrifuge at 20,000 times G for 2 minutes. Carefully remove the supernatant, leaving about 15 microliters behind so the pellet is not disturbed. The pellet may not be visible. Add 100 microliters of sample buffer. Mix by pipetting up and down 3 times. Heat the sample at 95 degrees Celsius for 5 minutes. Centrifuge at 14,000 times G for 2 minutes. Transfer the supernatant to a fresh 1.5 milliliter tube. The sample is now ready for qPCR setup, or it can be stored at -20 degrees Celsius for up to 2 weeks. For EP/JP or USP-aligned testing, extract DNA before amplification. The MagBeads Fast DNA Kit and the Spin Easy DNA Kit for forensics are validated for use with this test. When using the internal control as an extraction and process control, add it to the lysis buffer before starting DNA extraction. For the complete extraction workflow, follow the instruction manual for the selected DNA extraction kit. After extraction, the purified DNA is ready for reaction mix preparation. Prepare the reaction mix in a dedicated clean area, separate from the amplification zone. Do not bring post-amplification tubes back into this area. Prepare the required PCR tubes or wells for test samples, the no template control, the positive template control, and, when required for EP and JP workflows, the negative and positive extraction controls. If the internal control was not added before DNA extraction, prepare a 10-fold dilution. Add 5 microliters of internal control and 45 microliters of nuclease-free water to a fresh centrifuge tube. For each reaction, combine 15 microliters of 2 times qPCR master mix, 4 microliters of primer mix, and 1 microliter of 10-fold diluted internal control. If the internal control was added during extraction, use nuclease-free water instead. Mix thoroughly by pipetting up and down 5 to 10 times. Aliquot 20 microliters of reaction mix into each PCR well. Discard any remaining mix. Add the appropriate template to each well. Nuclease-free water for the NTC, 10 microliters of 10-fold diluted positive control for the PTC, and 10 microliters of DNA extract for each test sample and extraction control. Seal all tubes securely, mix well, and briefly centrifuge. Load the sealed PCR tubes into the qPCR instrument. In the software, assign FAM as the reporter channel for the Mycoplasma target. Assign VIC, HEX, or JOE as the reporter channel for the internal control. Program the instrument with an initial denaturation step at 95 degrees Celsius for 5 minutes. Then run 45 cycles of 95 degrees Celsius for 10 seconds and 60 degrees Celsius for 40 seconds. Collect data at the end of each cycle. After the run is complete, review all amplification plots and controls before interpreting the results. FAM signal with CT value below 40 indicates a Mycoplasma positive result. FAM CT of 40 or higher, with a valid internal control signal between CT 18 and 26, indicates a Mycoplasma negative result. If both FAM and internal control signals are absent, with CT values of 40 or higher, the result is invalid because PCR inhibition may be present. Repeat the run. Treat all amplified PCR products and samples as biohazards. Sterilize before disposal according to facility safety protocols. For technical support, contact the local MP Biomedical's technical support team or visit mpbio.com.